Ma'am, just click on that Monika's icon and you'll find all the
Monica
options
is
there.
host now. Now Monica is host now. I don't know how it is changing on its own.
Okay, no,
Now she can
no
share
issue.
it.
I'm really sorry to the gathering for all the trouble. Now I would again like to request Monica to invoke the Almighty.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमा आद्यां जगद्व्यापिनीं वीणा पुस्तकधारिणीम् अभयदा जाड्यान्धकारापहा हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्
May Lord Ganesha and Goddess Saraswati make this event a huge success. Further continue, if you have a dream, don't just sit there, get the courage to believe and leave no stone unturned to make it a reality. A dream does not become a reality through magic. It takes sweat, determination, and hard work. Finally, the most awaited moment for which we all have been working tirelessly since long. It's here. That's the inaugural of the Life Sciences Society for the session 2021-2022. On behalf of the entire Life Sciences family, I request our esteemed chief guest of the day and our honorable vice principal, sir, to please formally inaugurate the society since we are in the digital mode. So I would like to take the permission to follow the procedure virtually. Do I have the permission, sir?
Yes, go ahead.
Thank you, sir. So, ladies and gentlemen, here's presenting the official inauguration of Life Sciences Society for the session 2021 and 2022. Heartiest congratulations to entire Life Sciences family and everyone present here. We are grateful to our honorable principal, sir, for making this dream a reality. Sir has given us the path to follow and makes our dream reality and given the firm foundation for all the young minds for the incomparable hard work which he practices in daily life. Due to some administrative engagements, Sir is not present here, but with all due respect, we would now like to invite our Honorable Vice Principal, Sir. As someone said, there are no secrets to success. It is the result of preparation, hard work, and learning from the failure. The one who justifies the statement is our Honorable Vice Principal, Sir, Professor Kamal Kumar Gupta, Sir. Sir, I would like to request you to honor our Honorable Chief Guest of the day and address the gathering. Over to you, sir.
Thank you, Vikram. Thank you for your kind words. Good afternoon, everyone. This is a warm, bright, sunny afternoon, and same is the fate of Life Science Society, a bright, vibrant society. I feel proud and privileged to welcome the esteemed chief guest of today's function, Professor Rooplal. Sir, it's really a privileged moment for me that I'm welcoming you in the capacity of Vice Principal of Deshbandhu College. I welcome you in the capacity of Senior Most Faculty of Department of Zoology. And also, I welcome you as a student of yours. So it's really a proud and prestigious moment for me. I welcome Professor Rajiv Agarwal, Principal, who is very dynamic, always supporting, and who made this dream of Life Science Society true. I welcome Dr. Aditya Saxena, IQAC Coordinator, Dr. Indrakant Kumar Singh, DBT Star College Coordinator, all the participants, all the faculty members, all the students from Deshbandhu College and from other colleges, all the faculty members from other colleges. I welcome you all. At this point of time, I congratulate the Life Science Society, especially Dr. Manisha Sengar, founder, convener of the society, who envisioned this, pursued, and finally made this dream true with the kind consent of our principal, Rajiv Agarwal. Congratulate Dr. Manisha.
Thank you, sir.
Congratulate our other conveners, especially Dr. Roshni Rajamohan, Dr. Deepti Gupta, Dr. Kiran Bala, Dr. Anju Chibbar, and Dr. Vaishali Thakral. You all deserve a well appreciation and applaud to bring this Life Science Society to such a high extent. And also you have set such high aspiring goals just in a span of 1 year. And I can say in the college, this is one of the finest, one of the best society which is functioning. So congratulations and aspirations, my admiration to the entire team. This work wouldn't be possible without the energetic, bubbly, and aspiring students of the Life Science Society. There's a deep applaud to all of you. I congratulate on the second inaugural function. This is second inaugural function of Life Science Society. And I know you are going to come with flying colors. So this is about the society. Now, if I wish to say a few things about Professor Rooplal. Professor Rooplal is a person of international fame as far as the molecular biology is concerned. If I wish to say about the Department of Zoology, I can say one era was pre-Rooplal, and then there was an era of Professor Rooplal, where he really fly high and high without looking backward, without a stop. And he reached at an international level. Professor Rooplal's students are also doing wonders. Probably they are everywhere in all the colleges of Delhi University. If you go, they are everywhere in the topmost research lab in India, and they are among the topmost institute globally. So Professor Rooplal has given wings to his students to fly high and high. He was always supportive, kind, and persuasive. So whenever a student of Professor Ruplal come for interview, we never doubt about the credentials and potentials of that. We know that the student would have been trained well. A person of such like approachable, he is so simple. And I know he teaches, he's an excellent teacher. He teaches some of the very, very complex phenomena in such a simplified manner. Haste mazak karte, he have everything. So he's so simple that way. I remember I always boast of the things that I have a established research lab in Deshbandhu College. But let me share a secret this time. This lab was approved during the time when Professor Ruplal was head of the department. He supported and he encouraged me to establish my research lab. So that is a person of such a height he is. Now I will like only, I'll say only a couple of words about the title of the topic, because when Professor Ruplal is there, I think it is not wise to say anything much about it. But the title of the topic, who can be the best person to deliver a talk on this microbiome, human health, environment, and society. Now, whenever we talk about the microbiome, if I say we are living in the ocean of microbe, everywhere it is microbe are there in the entire biosphere. And we are in constant association of microbe, and a fact that if I say human body is made up of 30 trillion cells, but we harbor 39 trillion of microbes. So even the number of cells what our body have, we have more than that of microbe. So suppose if we commit some mistake, we can always say if we are not doing it's microbe inside us which are creating this mess. So we can always give all the negative vibes to the microbe. Microbes are the essential component. Whenever we talk about microbe, all of a sudden we say E. coli, we say disease-causing microbe. But microbes are maybe essential for the survival of us. They contribute at each and every place. Yeah, there are pathogenic microbes also, but yes, they are important for the very existence of us. But this is just one part of the story. And of course, there's entire story, entire facts, entire research we expect Professor Ruplal would be sharing with us. And one more thing I say, how popular he is. I joined this talk 10 minutes before, and that time there were 185 participants eagerly waiting for Professor Ruplal's talk. So he's so popular, and right now also on the screen it says 240, such a big number I've never seen on online. So without taking much of the time, I know everyone is eager to hear Professor Ruplal. So thank you, Vikram, and this is from my side. Thank you.
Thank you, sir.
Thank you so much, sir, for your words of wisdom. You have always inspired us. Optimism is the faith which will take you to the heights of the success. Now, I would like to invite, and I'm elated to welcome our coordinator, Life Sciences, Dr. Iringbam Rocky Mangancha, sir, to address the gathering and felicitate our honorable speaker for the day. Over to you, sir.
Thank you very much, Vikram. Honorable Chief Guest, Professor Ruplal, respected Principal Professor Rajiv Agrawal, Vice Principal Professor Kamal Kumar Gupta, and all the faculty members and students present today. I, on behalf of the Life Sciences Society, like to welcome you all. Thank you very much for taking out your precious time for this occasion. Today, I would also like to congratulate the founder convener of the Life Science Society, Synergia, Dr. Manisha Sengar, Department of Zoology, for taking up this huge initiative to nurture our students and make it successful. At the same time, I would like to appreciate the efforts of all the teachers and the student members of this society for putting all the hard work in making this inaugural ceremony a success. Professor Ruplal sir, I honestly like to admit that I was very much inspired by your teachings. Your lectures were full of knowledge and inspirations, and we were fortunate enough to be your students. In fact, most of the faculty members of our departments are your students. Sir, you helped our zoology department many times in different capacities, and we are proud to have you as mentor of our department in many occasions. Today, we are blessed again to have you in our inaugural ceremony of Synergia. Sir, we are very much excited to hear you, and thank you very much for all your blessings and support to our department and to the college itself. Thank you very much for coming, sir. Over to you, Vikram.
Thank you so much, sir, for your words of wisdom. Manika, I would like to request you, please make Indrakant sir co-host. He wants to say something.
Done.
Indrakant sir, you are co-host now. Sir?
Hello.
Yes, sir, you are audible, sir.
Yes.
You are
Oh,
audible.
yeah. Thank you. Thank you. No, please go ahead. Please go ahead. Yeah, nothing to say, sir. We are waiting for sir. Yeah, go ahead, please.
Okay. No issues, sir. Got it. So moving forward, now I would like to take the opportunity to invite Mahi Verma, a student of BSc Life Sciences second year, to formally introduce our speaker for the day.
Thank you, Vikram. It's my honor to introduce the guest speaker for the day, Professor Ruplal sir, whose contributions and achievements are immense, and some of which are as follows. He is the NASI Senior Scientist Platinum Jubilee Fellow at the Energy and Resources Institute, IHC Complex, New Delhi. He is the Fellow of Indian National Science Academy, New Delhi, Fellow of National Academy of Sciences, Allahabad, India, Fellow of National Academy of Agriculture Sciences, and the Fellow, Academy of Microbiological Sciences, India. He started his career as a lecturer in Sri Venkateswara College, University of Delhi, and served for nearly 13 years and became Professor of Molecular Biology at the Department of Zoology, University of Delhi, from where he superannuated. He has over 40 years long-standing experience in teaching, administration, and research. He has supervised nearly 75 PhD theses and mentored more than 120 MSc, MTech, and undergrad students. Professor Ruplal specialized himself in the area of microbial diversity, genomics, and metagenomics. Professor Lal was editor-in-chief of Indian Journal of Microbiology and also serving as a member of the editorial board in various leading scientific journals like M Systems, Environmental Microbiology, Environmental Microbiology Reports, BMC Biotechnology, BMC Biochemistry, and many more. He has over 240 publications in peer-reviewed journals with 8,618 ISI citations and H-Index 46. He has delivered more than 300 lectures in the area of biotechnology, microbiology, genomics, and metagenomics at various national and international platforms. Professor Ruplal was chairman of Board of Research Studies, head Department of Zoology, Dean Faculty of Science, Dean Examination, member of Executive Council and Academic Council, and member of several national and international bodies. Professor Lal is the recipient of several prestigious fellowships, including the Alexander von Humboldt Fellowship, Germany, DBT Overseas Fellowship, and Indo-US Professorship in Microbiology. He was a visiting scientist at the University of Cambridge, University of Kaiserslautern, Germany, Oregon State University, USA, EPFL, Switzerland, University of Lausanne, Switzerland, EAWAG, Zurich, Switzerland, CSIRO, Canberra, Australia, and Helmholtz Center for Environmental Research, Leipzig, Germany. He is the recipient of Lifetime Achievement Award by Association of Microbiologists of India, Endeavour Executive Fellowship, Mosellia Carter Distinguished Service Award from American Society for Microbiology, Professor S.R. Vyas Memorial Award, and Professor B.A. Johari Award on Microbial Diversity by Association of Microbiologists of India. He was the ambassador, American Society for Microbiology for the Indian Ocean region, and president of Association of Microbiologists of India. He was also a member of several national and international committees, a member of the review committee for Indo-US Professorship in Microbiology. He was General Secretary and is President of Indian Network for Soil Contamination Research. Presently, he is the ambassador of ISME, the Netherlands, to the Indian Ocean region. Sir, today indeed it's a pleasure that you have joined us today, and we hope that we'll get to learn a lot from your knowledge and experience on the topic. Over to you, sir.
Thank you very much for a very nice introduction. But before I move further, I would like to share the slides and probably. So, are you able to see my slides? Hello?
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
तो क्लियर है ना? आप मुझे
Yes.
थोड़ा बहुत ही ज़्यादा इंट्रोडक्शन हो गया, but anyway, thank you very much. At the outset, I must congratulate. I'm meeting online Professor Kamal Gupta since he became professor and vice principal of the college. My congratulations, and you really, the amount of hard work you had been doing in the college, you really deserved it. Also, Indrakant and many of my colleagues are working very hard and doing good amount of research in the college. It's not that you are doing good work, but also I know that doing the research in the college is really very difficult, because when I was in Sri Venkateswara College, I knew how difficult it is to do research in the college. So it's really very remarkable on the part of many of the colleagues, including Kamal, Indrakant, and many of you who are working very hard and doing good research in the Deshbandhu College. So my congratulations. I also congratulate Manisha, Kiran, Dr. Rocky, and many of the students who are really very enthusiastic and doing wonderful work while associated with this zoology department or life sciences department association. Once again, thank you very much for inviting me. In fact, all that has been said is not really true, but I accept you. You had been always my admirer, and that keeps me probably such lectures when I deliver and listen so high about myself. मेरी जिंदगी के कई साल बढ़ जाते हैं. तो कमल कह रहा था आप वैसे ही हैं, ये सब आप लोगों की वजह से है. एक्चुअली अब मेरी जिंदगी बढ़ाते रहते हो और ऐसे लगता है कि अभी भी मैं बूढ़ा नहीं हुआ हूँ. Although I superannuated in the year 2018. So let me start now a very important topic on which Kamal has already mentioned a couple of things. I will just spend more time on this. The microbiome, human health, environment, and society. Probably after superannuation, I thought that why not to actually, so that's why actually you will find in the title itself the society word, because I thought this is also right time for us to propagate microbiology. So there is this topic. I will not talk very high science, but would like to convince you and let you know, as Kamal has mentioned, that this is a very important topic and we all need to understand microbiome and microbiology for a variety of reasons. And the first thing which I want to mention, probably most of you know, being the students of life sciences, there is an unlimited world of microorganisms, and they almost live everywhere in the space, in the hot springs, in the ocean, and wherever actually you think of it, and you will find that microorganisms are already here. When I talk about microorganisms, I'm talking about a complete group of bacteria, archaea, fungi, protozoan, viruses that makes this entire group microorganisms. And as Kamal mentioned, there are something around when I'm talking to you, when I'm speaking to you, I'm interacting with some not less than 30 million of microorganisms. Even if a simple handshake leads to uh, 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 exchange of 124 million bacteria and it's something around 5.55 million bacteria. So that reflects actually how many bacteria are there. But how did you come to know actually that there are so many bacteria around us? They were there, but this was not really known to us. And this happened as a result of revolution in the sequencing technology and computational biology. Probably my colleagues, they know it now, that sequencing was very difficult sometime when we started doing it in 80s. Computational tools were not there, but the revolution has brought us to a state that we cannot do now any experimental work without actually sequencing whole organism or part of it or gene cluster and using computational biology. So this has again become very important. Probably this slide reflects that computational biology and sequencing has really become very important for even students who are doing, young students who are, let me make it very clear. You cannot now do good biology if you are not really a good, you are not aware of the computational biology tools. So would let you know in my subsequent slides that it is really very important for you to get a glimpse of this particular area and start thinking how best you can do or how you can get into this computational biology. Now, because of the revolution in sequencing technology and also in the availability of large number of computational tools, which are now freely available, large number of projects, they were started. Now, what I said that microorganisms or microbiology has become very important because everywhere there are microorganisms. That we came to know not by isolating these organisms, but by simply isolating the DNA, doing computational biology and making predictions that there are so many organisms around us. And as a result of that, sometime in 2006, large number of projects were started. Now, one of the important projects that was started in the year 2006 was Human Microbiome Project, immediately after the completion of Human Genome Project. So human microbiome, earth microbiome, there is a long list of very huge projects that has been initiated. But what these projects and sequencing technology and computational biology tells us, that there is an unlimited world of microorganisms, and the estimates are that number of microorganisms on this particular planet is something around 0.9 to 3.2 into 10 to the power 30 microbes, means bacteria, archaea, fungi, protozoans, and viruses. And viruses alone, the number is something around 1 into 10 to the power 31 viruses. And if you calculate the total living biomass on this particular planet, it comes to 27 to 30%. Nearly 30% of the living biomass is constituted by microorganisms. Be sure about it. So then we come to a question comes to my mind and to your mind. Can you do biology without understanding the role of microorganisms, which are constituting 30% of the total living biomass on this particular planet? Or can you live a good life without understanding the role of microorganisms? I think the answer to these questions, even the microbiology is not only important for those who are doing biology, it has also become important for those who are not biologists or even for our society. Now, when I talk about a microbiome, earlier we had been isolating single organisms and then looking into the microscope, doing all sorts of experiments with microorganisms. But now we select a particular niche, we isolate DNA and do the sequencing. And by doing computational biology, we look into a complete set of microorganisms that are present over there, the genomes that are present, and the genes and the functions, all these microorganisms, they are performing. So a collective term where you not only study the community of microorganisms, but we also study the genome and genes and the functions the microorganisms are performing. We call it as a microbiome. So that was a very important project in the year 2006, that was started, that is Human Microbiome Project. And today, as Kamal mentioned, the microbe being so important and abundant that our society is not aware of the benefits and havoc they can create. Now, probably you know it, all of it, but just I want to re-emphasize and again want to convince you that what I'm saying is really very important. Now, just imagine the weight of a single coronavirus, SARS-CoV-2, is something around 8.5 autograms. And the number of viruses that are needed to make a person sick is something 7 into 10 to the power 9, so 17 billion. And the total weight of those 17 billion viruses, it comes to 5 into 10 to the power minus 7 grams. And let's assume at one point of time, if there were 2 into 10 to the power 6 million of infections, or 2 millions of infections, the total number of the viruses, the weight of the viruses that infected 2 million people was less than 1 gram. So less than 1 gram weight of the virus or the number of the virus brought the entire planet on its knees. And the past 2.5 years, you will find everybody is sitting at home doing online work, and still we are not out of this particular problem. So imagine the amount, not that they are harmful, but what I'm telling, the havoc these viruses or bacteria, archaea, they can create. So this we need to understand. I mentioned earlier what is microbiota when you look only into the microorganisms, but when you are looking full complements of microbes, their genes, proteins, genomes, in a particular dynamic environment, we call it the microbiome. So those who are not familiar with this particular term, I'm re-emphasizing it time and again, and it has become very important. As a result of that, every year on 27th June, World Microbiome Day is celebrated, not only to propagate microbiology among biologists, but to propagate microbiology among the members of the society, in the public, to raise awareness. So that because of the role of microorganisms they are playing, this particular discipline, especially microbiome, has become very important. And as mentioned earlier, understanding microorganisms, I will come to this point later, you can lead a very good health. But give you one example. When in 2006, when this project on Human Microbiome project was initiated, its first phase was completed sometime in 2011. Now, what this Human Microbiome project reflected, that when we are born, we are 100% human. Our genes, our genome, 30,000, what you call it, is the number of genes in my body. But immediately afterward, as Kamal has also mentioned, that we are taken over by microorganisms, and 90% of the estimates are, of course, these are being checked and rechecked every time you find a new figure coming up, but large number of the genes that control our body, they come from microorganisms. And these microorganisms, in association with our body, they perform large number of functions with my own gene, which I cannot. Like 10% of the daily calories are derived from microbial genes. Mother's milk that contains glycan cannot be digested by my own enzymes. We need microbial enzymes. Vitamin B2, B12, folic acid are produced by, and they are not always harmful. They are also, that is helpful. So this number already mentioned, it keeps on varying. We are not exactly sure how many microorganisms are there in my body, but roughly this number is something around many times higher than my own cells. So that is very clear now. And as mentioned earlier in the introduction itself, Kamal mentioned that we are not now living, human beings are not alone. They live in association with a large number of organisms. So micro, human cells, microfungal associations is finally something that decides what type of individual I should be. And the maximum number, remember, the highest number of these microorganisms is in my gut, in the colon. Something around 10 to power 11 to 10 to power 12 colony forming units per gram of fecal matter. तो इतना ज्यादा नंबर है इनका, and they are not only, maximum number is in my gut, but they are almost present everywhere on all systems. So these figures, which again being mentioned or being taken up from the scientific literature, the point I wanted to tell you here is that each one of us is carrying something around 1.5 kg of microbial load. आप सभी actually 1.5 किलो आपके total microorganisms है, and this probably what do they do? They provide immunity. And immunity, in fact, actually compromising with the immune system would mean inviting a lot more diseases. The number of these microorganisms, which is very high, it varies from man to woman, infant, elders, obese, and also in a single human being, you will see the number is different on my skin, and the type of microorganisms that are present in my oral cavity or in my gut and almost everywhere. It depends actually. Now there are reports available which type of bacteria are predominant in the skin and what type of bacteria should be there, which are a dominant phylum in the this thing. And mentioned again, repeating it, that we are associated with microorganisms. There are 30 million bacteria around me when I'm speaking with you, when I'm talking, or when I'm sitting here in the room. And even you go to the ATM, you actually, the person who had come before you actually to draw the money, left some bacteria, and you actually take, so there is a swiping actually, taking bacteria actually from the, even from the ATM when you are withdrawing the money. But they are very important. And a human, more than this, there is a nexus between human brain and gut microbiome. In the gut, these microorganisms, they produce chemicals. And these chemicals through the nervous system are carried to the brain. And human gut microbiome is also known as second brain. I add a virtual endocrine organ. The composition of the second brain affects our first brain by a gut-brain nexus. So it's very important now. As I mentioned now, that 90% of our functions, directly or indirectly, are being controlled by the genes that we derive or that come from microorganisms. So that would mean any deviation from the healthy microbiome, from the healthy microbial population, which you call it a dysbiosis, would lead to large number of, believe it, it is so. And now people are, as I mentioned, that gut microbiota is responsible for maintaining our immune system, and any compromise lead to large number. I have listed only how many? Some 13 diseases, but there is a large number of diseases that are not listed here. Be it obesity or leanness, malnutrition, diabetes, heart diseases, blood pressure, depression. You take up any disease, almost all diseases are derived from like the disturbances in the microbial population, particularly in my gut. We did one study sometime in 2018, and that paper appeared on it says cover page of Environmental Microbiology, where we wanted to see actually the changes in the microbial population in the gut of TB patients, and also in the normal patients and those TB patients who are taken up antibiotics. So what we found actually, that there was an increase in the TB patients, there was an increase in the number of bacteria that produce short-chain fatty acid, and also that produce propionate and butyrate. And the production of short-chain fatty acid, propionate and butyrate, eventually known in the literature that it impairs immunity and also alters appetite. That is what happens actually in the tuberculosis patient. Just to give you one example, that any change in the gut microbiota or in the gut microbiome can lead to large number of, but this particular information that has come up has also got some application. I'm not getting into it. You can get into the, just give a Google search and you will find a lot more applications are coming up now. And doctors have really now treat human beings in association with microbiome microorganisms. So they also need to be trained actually to let them know very clearly the role microorganisms are playing in maintaining our good health in human beings. And there are now reports of fecal transplantation for a couple of diseases for which there is no cure. The scientists they have already worked out healthy human microbiome, replacing the defective microbiome and making human beings very healthy. So there are a large number of such reports already there, and probably wait for another 4 to 5 years. You will find a lot more application from human microbiome to treat those diseases which are in fact difficult to treat. You will have solution to those problems. Even there are companies that have come up, Open Biome, where they send you the kit and they are collecting the samples from healthy individuals, healthy microbiome, and storing it for subsequent use. And for collecting each sample from the healthy individuals, probably the companies, they pay you something around $50 per fecal sample. So what I wanted to tell you in a short time, that there are a large number of applications that are coming up, and many of the diseases for which even antibiotics were not there can now be treated by using information from microbiome. That's what I said actually. The first paper which I read, probably also would like to mention that what we did during lockdown, when after superannuation, how did we maintain a balance between professional and personal life? Kamal and many of my colleagues in the college, they have mentioned, I was not meant to do them, but I lived with them, and we enjoyed actually talking about science. And I know they are very brilliant and have born at a time when there is so much information and instrument and everything is available to them. So what we did actually, we made a group of some 16 students immediately after the lockdown in March 2020. And then we decided actually what to do while sitting at home. We decided large number of projects which I'll not mention, but one of the projects which we wanted to take over was the comparative genomics of SARS-CoV-2 coronavirus. And that time 95 genomes were available. We all sat together, did genome annotations, analysis, phylogenetic analysis, both pathogenic interactions and all sort of things. But what we concluded at that time, that paper got published in M System, that there are a large number of mutations that this particular virus is undergoing. And couple of mutations are making this particular virus more virulent. So every time you will find after 3 months or 4 months, new varieties or new variants of this virus are coming up. And not only this, we also looked into the host pathogen relationship. So not giving you much details of it, but what we concluded was that yes, this virus is very rapidly mutating, and couple of mutations that are being retained by the virus are making this more virulent. So as a result of that, you will find month after month or year after year, new variants of this particular virus, they are appearing. But what I wanted to convey that it's not only important for scientists to know microbiology or microbiome, but even your parents or the members of the society, if they have little bit knowledge of microbiology, and if you tell them that 90% of my functions are performed by microorganisms, you can really lead a very healthy life. In order to change your microbiota, make it healthy, make it normal, 3 things I believe and I have tried myself. You can change your lifestyle completely. You can change your food habits, stop taking antibiotics, but all these things you can only do when you are healthy. Once you are sick, you cannot actually start changing your lifestyle or start changing your. If you do these 3 things, I'll tell you, the first paper that I read, sometime probably Inder Kant and Kamal and many of you, they know it actually. that my weight sometime in 2010 or 11, it was around 97 kg. But when I read first paper that came out of human microbiome studies in Science in 2011, now that reflected that you are obese early, not because of your own gene, but basically because of the bacterial population that is present in your gut. And it has been studied actually that in obese individuals, the gut population, predominant bacteria belong to the phylum Firmicutes. They are fat lovers. And in lean individuals, there's another population of bacteria. So that means if you can change your fat lovers with carbohydrate lovers, you can, from obese, you can become lean. So from that paper, earlier probably I was thinking that everybody was telling me, I'm obese because of I eat more. But then later I realized my bacteria makes me to eat more, makes me to eat that stuff actually which contains a lot of fats. I loved gulab jamun, jalebi, and everything, samosa, so much at that time. But then I realized it is basically because of the bacterial population that is present over in my gut. So wanted to fool these bacteria and change my bacterial population to carbohydrate lovers. So it took almost 3 to 4 years for me. I changed completely my lifestyle, stopped eating actually. I do not eat chapati and rice actually. I simply eat once in a day. I take milk in the morning, do not eat much, but I take fruits, but I eat only once in a day, dinner time. Although I wanted to take my lunch, heavy lunch, and don't want to eat breakfast and dinner, but some of the compulsions in the family, I want to sit with them and enjoy eating, so we do it but prior to this, let me tell you very frankly, and that used to happen with me also. Whatever used to be left off in the evening, my wife will say, 2-3 चपाती बच गई हैं, फ्रिज में रखनी है, 2 सब्जी बच गई हैं, तो तुम क्यों इसको बर्बाद करना है, खा लो. That means literally my gut was used as a dustbin. I continue. Now I simply throw it off. I don't want actually to eat and then throw it off, but I don't eat now. I'm very strict. And as a result of that, I reduced my weight from 97 to something around 73. My blood pressure, which used to be something around 90, 170, it is around 75, 130. But then I walk around 15 kilometers a day, 21,000 steps, and then eat less, and I'm very active, and I also have a gym in my home, a cross trainer, where I spend almost 1 hour on the cross trainer. So that means you can lead a very healthy life just by changing your lifestyle, food habit. It takes only few weeks. It is difficult for you to immediately stop eating gulab jamun, jalebi, and all sort of things that makes you fat. But once you stop it for 10 to 20 days, or if you don't take it for 1 month, then you don't feel like eating. So that means changing lifestyle, changing your food habits. I used to take antibiotics. I had spondylitis. I have high blood pressure. I used to take a lot more medicines. Believe it, no, I don't take any medicine. When I will tell you a story actually. I went to a doctor and I was literally not able to climb this staircase. And told him, then he said, "Aapka jo grease hai na wo khatam ho gayi hai." And then I realized actually and started eating less and doing exercises, reduced my weight, and I'm so active now. And then again I met the same doctor. Ke bhai mera grease to fir aa gaya hai. Then he was surprised to see and said, "I want to come to your house and see exactly what you did and how did you become so active and normal." So that's what I'm telling you, that you go home and tell your parents the benefit of knowing actually microbiology. So this is one aspect I wanted to deal with it quickly. I will also let you know the microbiome and the environment. As I mentioned, that microorganisms are almost everywhere. We had been doing research with one of the site in Lucknow, where there was almost half of it was chemical put by the industry at a dump site. And we thought at one point of time that there will be no microorganisms. So that was first paper which we published on microbiome, metagenome. where you took the samples, isolated the DNA, and then actually did computational biology and make large number of predictions and found out that where the place where we were expecting that there should not be any organisms, there are large number of microbial community, and they are completely different, and most of them, they in fact eat those chemicals. Not only that, we found out that it's a very dynamic environment where there are catabolic genes, there are plasmids, there are IS elements, and so on and so forth. So just by you can plan. Now, based on all this, we have now planned actually to do field trials in order to do the bioremediation, because we know it will be a complete actually group of organisms or microbiome that we need in order to do bioremediation, and not a single organism. Another example I'm giving you where we worked is in Manikaran. Now, this is a very scenic place located in Kullu district of Himachal Pradesh, on the that is the Parvati bank of the Parvati River. And if you go there, you will find, you must go there. There hot water gushing out. It is around 96 degrees centigrade. It's a very scenic place, at the same time not very expensive. There is a Gurdwara, you can go there and they provide you free langar and also accommodation. So probably those who are not gone must visit and see. But what we did actually from the hot spring, from where the temperature was 96 degree, we isolated the DNA and did the sequencing, and then studied the microbiome. Now, we found out that even at this particular temperature, there are a large number of viruses present in this particular sample. At the same time, we also found out that although the normal bacterial populations are there, but some of the bacteria, they are highly enriched, like Enterobacter and Mediology. So we wanted to see why the population of Mediology is so high. And we looked into literature and found out that this Mediology is a bacterium that feeds on all gram-negative bacteria. And it simply enters into gram-negative bacteria and then multiplies at the expense of the bacterium. And immediately there is a lysis and new progeny of Mediolo is being actually released. So that's how actually the people have contemplated that Mediology can also be used as a live antibiotic. So what you did actually, we simply enriched the samples and then filtered the samples, enriched the Mediology and added E. coli. In order to see if Mediolo is there, it will sit on E. coli. So you can see in the next slide, Mediology, the small organism sitting on E. coli. It's not invading, but we could capture it sitting on E. coli probably, and found out that it does not invade, but sit on, this also we call it as epibiotic mode of predator-prey association. So not only this, because we also got an evidence that there are viruses at Hot Spring Manikaran, we assembled some 65 genomes of the viruses. We could also link them with the bacteria, and by doing enrichment studies, when you did this scanning electron microscopy, you will find these bodies sitting on bacteria, these small spots. So even your wet lab experiments, you can easily design by doing in silico studies. So moving beyond, we also took an adventurous expedition track further to Kheer Ganga. It was a very difficult track, and somehow we could reach an altitude of 3,000 meters above the sea level at Kheer Ganga. And here there is a sulphur spring. We isolated the bacteria, did the metagenome, and this study is already being published, where we found out that there are a large number of sulfur-eating bacteria, and probably by understanding this and isolating a couple of more bacteria, we can develop some application. So we did not stop over here. Currently, through our company, which is a small startup, we are also doing actually GIS-based mapping of microbial diversity across Ganges, along with a couple of other institutes where we have to do the bioinformatics. So we are collecting samples starting from Gangotri up to Diamond Harbor. The first phase of this particular project is already over. The second sampling, we are doing it, and probably in a year's time, we'll have more information to share with you what exactly. So this is, as I mentioned, and my colleagues also mentioned that we have some 75 PhD students who are doing very well in different areas as a teacher, as researchers, as students, as scientists at various places. So this human resource is really something which keeps me very healthy, and something which mission which we wanted to take up. Alone I cannot. I discussed with them that why not to propagate actually microbiology in this society, because even biologists are not completely aware of the benefits that you can derive from understanding the microorganisms that are associated with you. We have neglected the microorganisms or microbiology. I give you one example right in the beginning, that I told you actually that the weight of this particular single coronavirus is something around 8.5 autograms. And the total 2 million of population, if it has got to be impacted, then less than 1 gram can completely bring down this planet on its knees. So this is not known actually. Immediately after the lockdown, probably initially everybody was happy that it will be really a good opportunity to stay at home, but then the havoc this virus caused, everybody became very scary. So we have not told our children the potential of microorganisms, the potential of viruses, the benefits and the harm these microorganisms they can cause. Probably we have been only, and in fact it was a very difficult time for me to do microbiology in zoology department. We have always telling actually our kids, our children, and it is so even today, A for apple, B for banana, C for cherry, E for elephant. But we neglected, I mentioned right in the beginning, 30% of the living biomass is constituted by microorganisms. We never told our children A for Arthobacter, B for Bacillus, C for Corona, E for E. coli. They are not aware of the potential of these microorganisms. Even our students actually doing life sciences are not really aware of the potential benefits that it could drive. So in this direction, we are writing actually editorials. One of the editorial with 40 of the international scientists, where we were also a part of it, we have written it. It's very nice document. You can just search the literature and read it. The urgent need for microbiology literacy in the society. We have translated it into Hindi, Punjabi and Bangla. Another article, editorial, visualizing the invisible. That is for school children. We have written it. How to actually make them aware, how to make this exercise very impressive, to let them know that microorganisms, they are around us. We had been, along with all my colleagues, and in fact, students, we have been going to the village schools somewhere in Himachal Pradesh, a group of some 60 students, and then teaching actually school children, making them aware of the microbiology. They were very excited and they wanted to know actually the potential of these microorganisms, the antibiotic they produce. We could show them the nodulation, and we can show them actually that curd contain actually under microscope the bacteria and protozoans. We are also writing a complete actually issue in Indian Journal of Microbiology on microbial literacy. And recently, all my colleagues are group, those who were brilliant students and now they are teachers. We have written alphabet of elementary microbiology. This has appeared as an editorial in Indian Journal of Microbiology. So we are writing simple books that will be online available free of cost. Effort to taking microbiology to kindergarten or to graduate level or to the society. Not only this is also being done in Hindi, so that we do our something which is missing. Not only our society need to know about microorganisms, but even the administrators actually, the governments actually, while making the policies, they need to now actually understand the role of microorganisms. As I mentioned initially, we started some 30 to 40 of my colleagues are students, but now many people from international platforms and institutes, they have joined us, and we are taking it, we are becoming a part of International Microbial Literacy Initiative, where we want to take it now propagate microbiology, not literacy, not only in India, in the society, but also we want to propagate. So every time when we organize a conference, a workshop, we keep a complete session on microbial literacy. Even along with our colleagues, we have organized International Microorganism Day, Microsphere, on 17th November, September of every year. This is being done. A conference that was organized by us, we actually kept a special session on microbial literacy, propagation of, where we invited formal school children, member of the society. Even the calendar that I just released, actually, it is based on microbiology. And most of the work we are doing through our colleagues as well as a small startup where my actually students are helping me to do bioinformatics, to train actually young graduate students and so on and so forth. So this is another workshop we have planned from 1st to 2nd March, again on microbial literacy, children microbial literacy outreach program. Probably we will be conducting in the first week of March. But with this, I wanted you to become my ambassador. The purpose of this lecture was to let you know and to make you, to convince you that microbiology is really very important. So you please, when you become my ambassador, once you go home, tell your parents the role of microbiology, tell your sisters, tell your children, everybody, that you need to understand the role of microorganisms, not only to maintain good health, but a good nation, healthy nation actually can only be attained by understanding, propagating this particular microbiology among society and children, and introducing actually microbiology right from the school level. So tell your parents not to take antibiotics if they are right now, if they are at the age of 60, 62 or 60, they should start actually changing their lifestyle and leading a normal, healthy life. So my request is, please become my ambassador, propagate microbiology almost among society members, which is really and urgently needed. Thank you very much.
Thank you, sir. Greetings, everybody. I am Arushi Dogra, Secretary Synergia. Sir, we thank you from the bottom of our heart for taking time from your busy schedule to be the guest speaker at our function and delivering such an apprehensive talk. The strength of around 300 participants is quite evident of the fact how insightful your lecture was. Your presence and wise words helped magnify our cause in best possible way. Now I would like to request you to entertain few queries from our audience. Can we proceed, sir?
Yeah.
Sir, first question is from Surya. Sir, as the microbes came first in our nature, do you think they are simply the results of trials and errors made by the nature in order to instill the population on Earth?
Pardon, can you repeat the question?
Sir, the question is, as the microbes came first in our nature, do you think they are simply the results of trials and errors made by the nature in order to instill the population?
Probably is it a question or you want to answer for me? I could not really understand what is that you want me to tell you about
Sir,
it.
the question.
Yeah.
Sorry to interrupt, sir, but the question is basically that these microbes are the result of the mistakes made by the nature.
Oh, I don't know whether you call it as mistake or you call it as evolution. I mean, these microorganisms, yes, we know many billions of years, the first life that appeared actually on this particular earth were microbes. They were then anaerobes. And after that, probably what I gave you the example of microorganisms staying in the hot springs itself reflects that the first living organisms that were to appear were microorganisms, and even the mistakes that probably nature did, now we can also do those mistakes. People, and actually Craig Venter, he constructed a complete functional nucleus with minimum number of genes that are needed in order to replicate a cell. Only thing scientists could not do was could not create a sort of membrane and cytoplasm. Now, once that also becomes possible, probably we will be able to create a life by, to some extent, we will be able to understand how nature created this particular life. But how it was done, it is still a mystery.
Okay, thank you, sir. So, next question is from Shivang, who is asking, why is our normal flora so important from immunological point of view?
Because, as I mentioned, that the immunity, basically you are healthy because, for example, you have, you probably now know it, that in a family where everybody was infected with the coronavirus, but there were one or two individuals or one person in the family, he was normal. He was not infected. So that means the immunity decided actually how immune you are will decide whether you will be. Same person, you eat outside actually, probably nothing will happen to you, but somebody else eating the same food will have a lot of problems. So microorganisms, through chemicals which they produce in my gut, the signals go to my brain, and the good organisms will actually, as I mentioned, did not let you know got into the details that these microorganisms primarily, इधर से सब सारी चीजों से घुमाने के बाद ये आ जाता है कि हमारी इम्यूनिटी जो है वो माइक्रो ऑर्गेनिज्म पे डिपेंडेंट है. अगर मेरा हेल्दी पॉपुलेशन होगा तो मेरी इम्यूनिटी होगी. Those organisms will not allow pathogens, all bacteria, all type of bacteria, all pathogens, Mycobacterium, everything is there in my gut, but they are in very low population. And the healthy organisms, microorganisms, do not allow them to propagate. तो जो immunity की बात है वो इन्हीं से आता है, सारा बैक्टीरिया से. And immunity, in fact, कोई भी disease किसी को हो जाता है तो basically immunity कम होती है ना, इसी वजह से सारी problems है.
Yes, sir. So next question is from Sapna. She is asking, how does pollution affect the human microbiome, and are the impacts equally distributed?
देखो, पॉल्यूशन की तो बड़ा छोटा फैक्टर है. आप कहाँ पर रहते हो? कमला नगर में रहते हो, वहां पर गुलाब जामुन खा रहे हो, है ना, ऑयली फूड खा रहे हो, आप जलेबी खा रहे हो. सो ऑल दिस कंप्लीटली डिपेंडेंट अपॉन पॉल्यूशन. तो एक बड़ी छोटी सी बात है, पर आपका जो सारा माइक्रोबायोम है वो आप क्या खा रहे हो उस पर मेजर इसके ऊपर इम्पैक्ट है. अब व्हाट यू, ऑफ कोर्स एवरी एस्पेक्ट, इवन मसाज, इवन वॉकिंग, एवरीथिंग इम्पैक्ट्स योर माइक्रोबायोम. बट मोर देन दैट, आपका ईटिंग हैबिट्स जो है, आप डेली क्या चपातियां खा रहे हैं या आप इडली डोसा खा रहे हैं. Certainly now there are studies available where they reflect कि भाई माइक्रोबायोम आपका ज्यादा खाने की हैबिट से चेंज होता है. बट ऑल अदर, दे आर माइनर, बट दे ऑल अदर फैक्टर्स, पॉल्यूशन, योर लाइफस्टाइल, योर एसोसिएशन विद व्हाटएवर इट इज, आप कैसे हो, आप झगड़ा करते हो, लड़ाई करते हो. इनफैक्ट मेरे घर में हम बहुत लड़ते थे, हस्बैंड एंड वाइफ. बाद में पता लगा ये बैक्टीरिया की वजह से है. अब कोई लड़ाई नहीं होती है. तो ये सब कुछ ही बैक्टीरिया से वजह से है. ये मान लो आप 90% मैंने ऑलमोस्ट नियरली कमिंग टू 100%. हमारा तो अपना कुछ भी नहीं है, सारा बैक्टीरिया ही बेवकूफ बना रहे हैं हमें.
Okay, sir.
Yeah.
Yes, sir. So next question is from Khyati. She is asking, sir, what are the future perspectives of microbiology?
देखो, वो मैं ये बता दूं कि बहुत अच्छे हैं. मैं जूलॉजी करके भी ठीक ठाक रहा है ना. Yes, microbiology, it has got a future. Otherwise also you don't take microbiology, you take zoology or botany or life sciences में आपको माइक्रो ऑर्गेनिज्म से ही शुरू, बिकॉज आई सेड इतना ज्यादा एसोसिएटेड हैं तो माइक्रोबायोलॉजी तो करना ही होगा. नॉट दैट यू नीड टू बिकम एक्सपर्ट इन and identifying the microorganisms, but you need to become expert in computational biology. Computational biology आपको सीखना है जो कॉलेजेस में नहीं है. आप कहीं से भी C++, पाइथन, कंप्यूटर सीख लो, बहुत सही हो जाओगे. कुछ भी करो, फाइनली if you are good in computational biology you can do wonders, and this is the time for you. आपका तो बहुत लंबा जिंदगी है, आपने तो शुरुआत ही की है. अगर मिस कर जाओगे, probably this is not being taught in the colleges, but उसको वेट करोगे तो फिर आपको you will be losing something which later you will realize. अब मैं रियलाइज करता हूँ कि अगर मुझे कंप्यूटर आता होता तो मैं कितना सुखी होता, कितना अच्छी अच्छी चीजें कर लेता. बिकॉज माई कोलीग्स नोइंग लिटिल बिट ऑफ कंप्यूटेशनल बायोलॉजी, दे कैन डू माइक्रोबायोलॉजी. द अदर थिंग एडवांटेज इज माइक्रोबायोलॉजी करने से ये सिंपल सिस्टम है. यहाँ से जीनोम का या एनोटेशन शुरू करना, जीनोम साइज बड़ा छोटा है. कंप्यूटेशनल बायोलॉजी सिंपल एग्जांपल्स हैं, मटेरियल बहुत इजीली अवेलेबल है. तो बट कितना स्कोप है इसका वो आपके अपने ऊपर डिपेंड करता है. आप कितने अच्छे हो वो सबसे ज्यादा मैटर करता है. इस वजह से नहीं, आप माइक्रोबायोलॉजी लेके भी बहुत फ्रस्ट्रेशन होगी. जिंदगी इतनी आसान नहीं है. हमें पता है ना कि आपके कोलीग्स जो आपको बोल रहे हैं, ये बोलते हैं बहुत अच्छा स्कोप है. बेटे नहीं है, हिम्मत चाहिए. आगे तुम्हारे से भी बहुत अच्छे अच्छे बच्चे हैं, आपको compete करना है. सो स्कोप तो आपने बनाना है. बट यस, माइक्रोबायोलॉजी इज समथिंग इनकमिंग सब्जेक्ट which has got tremendous application.
Yes, sir. So next question is from Bharti. She is asking that she has heard about magnetotactic bacteria that can be used in future for database storage. How can it be possible, sir?
You know, देखो पहले मेरे लिए भी सरप्राइज था कि क्या ये जो सब कुछ कर रहे हैं, बैक्टीरिया इलेक्ट्रिसिटी बनाते हैं, है ना. तो ये बैक्टीरिया को भी, बैक्टीरिया के अंदर नैनो पार्टिकल्स है ना. अगर आप कोई चिप बना लिया और बैक्टीरिया में डाल दिया या कुछ भी स्टोरेज की, डाटा स्टोरेज की बात आ रही है. नाउ आई थिंक आपको ये पता नहीं है कि बैक्टीरिया को हम लाइफोलाइज करके करोड़ों कितने भी साल रखना है रख लो, कुछ नहीं होगा रूम टेम्परेचर. या उनको -80 पे आप स्टोर कर सकते हो. तो अगर कोई डाटा अगर हम किसी फॉर्म में बैक्टीरिया में, नैनो पार्टिकल के फॉर्म में बैक्टीरिया में डाल दें और उनको फ्रीज कर दें तो डाटा तो स्टोर हो गया, वो खराब ही नहीं होगा. हम फिर रिवाइव कर लेंगे, है ना. तो दे कैन बी, दे कुड बी, माने जो आप सोचते भी नहीं हो वो एप्लीकेशंस आप बैक्टीरिया से, हम बैक्टीरिया को पहले इतना नहीं सोचते थे बट दे आर आल्सो. अब तुमने सोचा था कि पूरी दुनिया को एक वायरस जो है वो घर में बिठा देगा? तो ये तो बड़ी छोटी सी बात है डाटा स्टोर करने की बैक्टीरिया के लिए. जब वो इतना बेवकूफ आप सारों को बना सकते हैं ना, तो ये डाटा तो उनके पास होगा ही, है ना. राइट, इट इज पॉसिबल. बट डोंट अंडर एस्टिमेट माइक्रो ऑर्गेनिज्म्स. ये मत कहो कि वो सिंपल एंड फॉर्म्स है, वो बहुत बहुत ही चालाक है. वो हमारे से भी चालाक है, अभी भी हमारी जान नहीं छोड़ रहे.
Yes, sir. So next question is from Rachna. She is asking that what are the effects of common food additives and preservatives on our gut microbiota?
ये सारा जो एग्जैक्टली अगर आप नेट में लिख दो कि ये खाने से क्या फर्क पड़ेगा, बट मैंने जो देखा है कि जो सर्टनली एज आई मेंशन आपके फूड हैबिट से ही, भाई हर एक बैक्टीरिया की रिक्वायरमेंट अलग अलग है ना. तो अगर मैं, मेरे बैक्टीरिया में फैट लविंग बैक्टीरिया है, मैं नहीं खाऊंगा तो थोड़ा दिन तो वो करेंगे गुजारा, बाद में मर जाएंगे. दूसरे वाले जो है ओवर पॉपुलेट करेंगे. So the requirements, what type of food is needed for bacteria, these are also different. That's why we are not able to culture all the bacteria. ये जो हम बोल रहे हैं ना कि इतने 0.9 into 10 to power 31 या 32 बैक्टीरिया हैं, वो हमें पता ही नहीं था. हमने तो 0.1% भी नहीं पता किया था. तो ये जो सारा आप जो खाते हो, certainly it makes a lot of difference in the population that I will harbor. But best thing ये होता है कि अगर आप कम खाओगे तो बैक्टीरिया कम डाइजेस्ट करेंगे, तो वो अगर आपको कुछ केमिकल बनाते हैं, हार्मफुल हैं, कोई लेवल बढ़ रहा है आपका, जब आप होल बॉडी चेकअप करा रहे हो, जब आपने खाया ही कम है, बैक्टीरिया को मौका ही नहीं दिया, तो फाइनली आप हेल्दी होंगे. बट सर्टनली, और एज आई मेंशन, all factors, almost everything, some of them are very predominant, the others are not so significant, but they impact the microbial population, especially in my gut.
Yes, sir. So next question is from Ishan. He's asking, is there any specific reason that why microbes are more concentrated in our gut?
You know, भाई आप भी जहां पर सब कुछ सुविधाएं अवेलेबल है ना, आप दिल्ली आ गए, मैं भी दिल्ली आ गया, क्योंकि यहाँ जो चीजें मिल सकती थी वो मुझे गांव में नहीं मिल रही थी जहां पर मैं रह रहा था, है ना. यहाँ पढ़ने के लिए अच्छे, तो गट जो है एक ऐसा, गट मीन्स इंटेस्टाइन ही एक ऐसी जगह है जहां पर बैक्टीरिया के लिए वो सभी सुविधाएं हैं, एनारोबिक बैक्टीरिया हैं, सब कुछ उनको मिल जाता है, स्पेस मिल जाता है, फूड, भाई जो कुछ भी तरह तरह की वैरायटी जो आप खाओगे, बैक्टीरिया भी तो खा रहे हैं ना. तो मेरे हाथ पे ज्यादा बैक्टीरिया बैठ जाएं, वो क्या करेंगे, आपने दूसरे दिन उनको धो देना है. लेकिन जो गट में होंगे ना, वो तो बड़े आराम से भी वो भी वेट कर रहे हैं कि आज गुलाब जामुन आएगा कल, है ना. सो दे ऑल्सो नो इट, कहाँ पर क्या चीज, कंफर्ट कहाँ पर अवेलेबल है, वहीं रह रहे हैं. जैसे आप भी जानते हो कहाँ पर कंफर्ट है, आप वहीं चले जाते हो सारे. Why Delhi is overpopulated? Because of so many things. So same is the thing with our gut, because many things that bacteria like are available for them to stay over there.
Yes, sir. So the last question is, microbes like tardigrades can survive in vacuum spaces also. How can they survive in harsh conditions like vacuum?
देखो, ये सिंपलर जीनोम है इनका, ये म्यूटेट बड़ा फास्ट होते हैं, है ना. अब आपको पता है कि जो ये थर्मस है, विच वी आइसोलेटेड फ्रॉम मणिकरण, जो थर्मस कैटिकस था, that was isolated from Yellowstone hot springs in USA. तो वो जो टाक डीएनए पॉलीमरेज जो बनाया था, वो 70-80 डिग्री पे भी सरवाइव कर जाता है. नहीं तो ये आरटीपीसीआर और पीसीआर जो करते हैं आप, अगर नॉर्मल एंजाइम करते तो वो एंजाइम जो है वो क्या होना था, उसको डिग्रेड हो जाना, दोबारा फिर डालना. मैं यही काम करता रहता. अब मैं वही एंजाइम डालता हूँ, सो आई हीट द सैंपल, डीनेचर द डीएनए एंड टाक डीएनए पॉलीमरेज जो मैंने थर्मस से लिया, उसको कुछ नहीं होता है. तो ये बैक्टीरिया जो है बहुत फास्ट अडेप्ट कर लेते हैं उन इन्वायरमेंट को जो बहुत ही हार्श है अपने रहने का. अगर कुछ भी नहीं हो तो ये स्पोर फॉर्मेशन या ऐसे ही लगेंगे मर गए. मैं कह रहा हूँ 80 डिग्री पे ये रह जाएंगे, कुछ नहीं करेंगे, पड़े रहेंगे. आप ऐसा तो नहीं कर सकते आप लोग, है ना, कि मैं कुछ नहीं खाऊं और 5-10 दिन में हो लोगे. लेकिन बैक्टीरिया कैन सर्वाइव. They have the ability to survive under any conditions, और बड़ी जल्दी म्यूटेट हो जाते हैं. बैक्टीरिया में ही तो प्लाज्मिड है, बैक्टीरिया में ही तो IS एलिमेंट है. तो वो जो इन सिचुएशन को सेंस करते हैं, उसी हिसाब से फिर बोल देते हैं बैक्टीरिया को, अब ये बनाना शुरू, पहले ही तैयार हो जाओ, which we cannot do it. We are more specialized.
Yes, sir. Thank you, sir, for answering all the queries with fortitude. Now, I would like to call our respected, magnificent, and diligent co-convener from Zoology Department, Dr. Kiran Bala, ma'am, to felicitate our honorable speaker.
Thank you, Rishi. Very good evening to everyone. Sir, it gives me immense pleasure in being your PhD student to express my special appreciation to you, sir. I, Dr. Kiran Bala, on behalf of the Life Sciences Society, Deshbandhu College, would like to extend my sincere thanks and gratitude to our eminent speaker, Professor Ruplal, sir, for accepting our invitations and to deliver the excellent talk on microbiome, human health, environment, and society. The talk was really interesting and enriching, and I am sure the participants have enjoyed it thoroughly. Sir, as a humble gestures, we are present a memento. Wait a moment. Sir, as a humble gestures, we present you a memento for being a speaker today and imparting your valuable insights. Please accept, sir. And thank you so much, sir, for being a speaker today and for being with us here.
Thank you very much. Thank you.
Thank you, sir. Thank you. Sir, it was such a wonderful lecture, sir. Itna informative or itna, and thank you, sir. You have to leave tomorrow and still you agreed. I'm really honored. Sir, we all are really honored because आप इतने बिजी हैं और कल आपको फ्लाइट भी है आपकी और आप कैसे आज मैनेज कर पा रहे हैं. इट्स रियली वेरी, वी ऑल आर रियली थैंकफुल सर. और बहुत बढ़िया लेक्चर. आई थिंक आपके लेक्चर के बाद सब लोग अपने ईटिंग हैबिट्स पे जरूर ध्यान देंगे सर. और सर एक क्वेश्चन मेरा था सर, ये जितने भी कैंसर्स हैं सब ये इतनी प्रॉब्लम्स हो रही हैं, जहाँ आजकल नई बीमारियां भी आ रही है और ऑटो इम्यून जो आजकल बहुत मेजर इशू बन गया है सर. जितने भी ऑटो इम्यून डिसऑर्डर्स हैं और कैंसर्स, ये दो बहुत बड़ी प्रॉब्लम्स हैं. और ऑटो इम्यून तो सर पहले इतना कम सुनने में आता था और आजकल तो हर चौथे बंदे को स्किन प्रॉब्लम है, कोई और प्रॉब्लम है, ऑटो इम्यून डिसऑर्डर्स हैं. कुछ लोगों को इतने फास्ट एनाफाइलैक्टिक शॉक्स लग रहे हैं, इतना फ्रीक्वेंसी बढ़ गई सर. ये भी सारा माइक्रोबायोम की
मनीषा, मैंने ये बताया, देखो अगर 90% जीन जो है वो बैक्टीरिया के हैं तो मेरा कोई ना कोई पाथवे कहीं ना कहीं जाके बैक्टीरियल जीन से हिट हो रहा है. और वो जो ये सब कुछ क्या है एक्चुअली, इट्स ए केमिकल रिस्पॉन्स. तो जो बैक्टीरिया फिर डिग्रेड करके जो केमिकल बना रहे हैं वो मेरा सिग्नल जा रहे हैं ब्रेन से, ब्रेन अकॉर्डिंगली मेरा रिस्पॉन्सिबल फॉर कंट्रोलिंग माय सिस्टम. तो सारी डिजीजेज डायरेक्टली और इनडायरेक्टली नाउ हैव बीन रिलेटेड टू द माइक्रोबियल. इनफैक्ट इवन आपने सुना होगा ये कोरोना वायरस पे भी कितना इम्पैक्ट हुआ है गट माइक्रोबायोम पे, है ना. तो सर हर चीज जब हम कह रहे हैं कि 90% बैक्टीरियल जीन है, दैट मीन्स कोई भी डिजीज है. आपको विश्वास नहीं होगा, आपकी साइकोलॉजी है ना, आप चिड़चिड़ा स्वभाव है आपका, आप कैसे हैं, आप कैसे कोई बंदा जो है उसको कोई फर्क नहीं पड़ता है, वो हंसता ही रहता है, दूसरा जो है वो बड़ा नाराज हो जाएगा. ये सब कुछ बेसिकली केमिकल रिस्पॉन्स है ना मेरी बॉडी, है ना. तो वो सारा कुछ हो कैसे रहा है, वो बैक्टीरियल, मेरे गट बैक्टीरिया जो है वो केमिकल प्रोड्यूस कर रहे हैं, सिग्नल ब्रेन में जा रहा है, अकॉर्डिंगली मेरा सारा मेटाबॉलिक पाथवे जो है इनडायरेक्टली डायरेक्टली बैक्टीरिया कंट्रोल कर रहे हैं. एंड इट ऑल डिपेंड्स व्हाट आई ईट एंड हाउ डू आई लिव.
Yes, sir.
But thank you very much. It was pleasure being here with you all.
One query tha sir.
Yeah.
Is there any program, short-term program for undergraduate students?
Yeah. We have here. We are running. Noida mein hi. They can write to me ruplal@gmail.com.
Okay, sir.
And I will be happy because in addition to propagating microbiology, we also want to feel very happy to train young people in this particular discipline as a part of our mandate actually to take this microbiology to young students and to the society. तो इस पे लिख देना आप ट्रेनिंग लेने के लिए. We can help them. We are free. हमारे यहाँ कोई बहुत रूल रेगुलेशन नहीं होते हैं. हम वो आते हैं, उनको काम सिखा देंगे, सीख लेंगे, तो कोई प्रॉब्लम नहीं है. राइट.
Thank you very much, sir.
सर थैंक यू सो मच. रूपलाल सर, इंद्रकांत बोल रहा हूँ. सर आपके आशीर्वाद से एकलव्य की तरह कुछ सीखा, कुछ नहीं सीखा. आपको गुरुदक्षिणा तो कुछ नहीं दिया, बट काम अच्छा करने की कोशिश कर रहा हूँ.
नहीं, आप बहुत अच्छा कर रहे हैं. आई कीप ए ट्रैक ऑफ योर प्रोग्रेस. यू आर डूइंग वेरी वेल इन टर्न माय बेस्ट विशेस.
और सारा कुछ सर आपके लाभ में ही छुप के सीखा.
अरे ऐसा कुछ नहीं है,
आपके आशीर्वाद से
वैसे बोल देते हैं, ऐसा कोई बात नहीं है. ये तो आपकी मेहनत है, दैट यू आर डूइंग सो वेल.
नहीं सर, थैंक यू सो मच. और बाकी सर स्टिल कई सारी चीजें अभी भी हैं जो सीखना है आपसे. तो वो सीखते रहेंगे। थैंक यू सो
Thank
मच।
you. Thank
जो
you.
आपने कॉलेज से रिसर्च एस्टैब्लिश करने के जो मंत्र दिए थे, कोशिश जारी है, कहाँ तक पहुंचेंगे, बस आपका आशीर्वाद रहना चाहिए।
बहुत आगे जाओ।
थैंक
Be
यू
sure.
सो मच,
Yeah.
थैंक यू सो मच।
Thank you, sir. It is always such a fascinating and interesting talk. As always, you know, listening you is always a treat for all of us.
नहीं ऐसा नहीं
Really
कमल,
wonderful. Thank you,
आप
sir.
Yes,
खैर
sir.
आप बहुत अच्छा बोलते हैं तो हमारी और जिंदगी बढ़ जाती है। थैंक यू वेरी मच।
Thank you, sir.
थैंक यू।
Thank you, sir.
Yes, sir.
Now, I would like to welcome Ankur, editor Synergia, to introduce our marvelous conveners and co-conveners.
Thank you, Arushi. Coming together is a beginning, keeping together is success, and working together is progress. Good afternoon, everybody. This is Ankur Bhardwaj, editor, Synergia, the Life Science Society of Deshbandhu College. And it's a matter of immense pride for me to anchor before you all on this auspicious day of inaugural ceremony. Empowerment isn't a buzzword among leadership gurus. It's a proven technique where leaders bestow their teams with enormous values, invaluable experience, and path-breaking guidance to yield the sweet fruits of success. Synergia, being the amalgamation of three disciplines, that is botany, zoology, and chemistry, stands for unity in diversity, ease in complexities, and togetherness in differences. And the one who gives expression to these amazing qualities are the working hands of Synergia. So now is the time to witness adroitness of Synergia. So let me take the privilege and honor to you all to introduce to you all Team Synergia for this year. First in the list of intellectuals is extremely dynamic, perspicacious, and insightful. Our life science coordinator, Dr. Irangbam Rocky Magencha, Assistant Professor, Department of Zoology, Deshbandhu College. Sir, your work ethics are amazing. Thank you for being part of Team Synergia. Following him is extremely skillful, assiduous, and dedicated convener, Dr. Roshni Rajamohan Mathur Ma'am, Associate Professor, Department of Botany, Deshbandhu College. Ma'am, you are a candle in the darkness for the society. Thank you for being a part of Team Synergia. Next is most ardent, accomplished, and dexterous convener, Dr. Manisha Sengar Ma'am, Associate Professor, Department of Zoology, Deshbandhu College. Ma'am, your guidance has always been the center narrative for Synergia. Thank you for being associated to the society. Next is most zealous, diligent, and talented convener, Dr. Deepti Gupta Ma'am, Assistant Professor, Department of Chemistry, Deshbandhu College. Ma'am, your suggestions are pole star for Synergia. Thank you for being part of it. Following her is most committed, fervent, and devoted co-convener, Dr. Anju Chibbar Ma'am, Assistant Professor, Department of Botany, Deshbandhu College. Ma'am, your inputs are invaluable, and thank you for being part of Team Synergia. Next is magnificent, brilliant, and dazzling co-convener, Dr. Vishali Arora Ma'am, Assistant Professor, Department of Chemistry, Deshbandhu College. Ma'am, your unique perspective and ideas have no comparison. Thank you for being associated to Synergia. Following her is dedicated, efficient, and worthy co-convener, Dr. Kiran Bala Ma'am, Assistant Professor, Department of Zoology, Deshbandhu College. Ma'am, your work ethics are amazing, and you stand with us through thick and thin. Thank you for being associated to Synergia. Now I would like to request Nupur, Council Member of Synergia, to take further the event. Over to you, Nupur.
Thank you, Ankur. Science may set limits to knowledge, but should not set limits to imagination. Life Sciences Fraternity, under the flagship of Department of Botany, Chemistry, and Zoology, constituted Life Sciences Society Synergia. Founded by Dr. Manisha Sengar, Associate Professor, Department of Zoology, Deshbandhu College, University of Delhi. Our dynamic principal, sir, left no stone unturned and made possible what seemed impossible by establishing this prestigious society. Its goal is to create and maintain a vital platform to enable budding life scientists to have a broad view of applying the three disciplines, that is, botany, chemistry, and zoology, together in fields of research, development, and education. It also aims on understanding social, ethical, and legal dimensions of the most dynamic branches of life sciences. Synergia, run by its very dynamic office bearers, council members, under the guidance of conveners, co-conveners from Department of Botany, Chemistry, and Zoology. No matter what work, it needs a team to function well balanced and put equal efforts. And no matter how hardworking the team members are, it needs a leader or idol to supervise them and to inspire them. I would take immense pleasure to introduce the diligent office bearers, council members, and class representatives who work day and night to carry forward the scheme of the society. Our president, Vikram Kamlesh Sehrawat, the most foremost, diligent, and modest member of the society, our Riduvan's best president. Introducing the innovative, imaginative, and dedicated vice president of the society, Ansh Sharma. The one giving equally innovative ideas and has a spirit of working selflessly for the society, the secretary Arushi Dogra. The joint secretary Samaira Bhutani, who is full of capabilities and forte. She's briefed to her responsibilities. Now I would like to call Tanishka Puri to take the event forward. Thank you.
Greetings, everybody. I am Tanishka Puri, Council Member of Synergia, introducing office bearers further. Manika Chauhan, our cultural head, is such a proficient and creative person handling all the cultural activities with grace. The editor of our team, Ankur Bharadwaj, a vocabularian and well-balanced personality, and is always ready to help everyone. With her innovative ideas and socializing skills, we have our media and PR head, Mahi Verma, such a flair personality. And last but not the least, we have our creative and intellectual IT head, Nyashi Srivastav, the amicable individual. Thank you for all the pain you take to keep our society afloat and to inspire us to be dedicated with our work. Your work ethics and your gracefulness are truly alluring. Innovations is taking two things that already exist and putting them together in a new way. Now, wholeheartedly introducing the other important members of the team, the council members of Synergia, who are working hard to give their best to take the society towards more success. The third year members are Aditi Rohila, Sapna Chaudhary, Sadhna Gupta, Alia Khan, Anamika Singh, Mehak Dhal, Kashish. The second year members are Shivang Malik, Varda Parveen, Rachel Regas, Mansi Chawla, Tripti Rawat, Sujal Sahu, and Suryadev Singh. The first year members are Ansh Bhatnagar, Nupur Singh, Khyati, myself Tanishka Puri, Rachna Prajapati, and Priya. I aspire to be as sincere and devotional as you are in your duties. Thank you for being kind enough to take the responsibility of being council members and working selflessly for the society. Quality performance starts with a positive attitude. The class representatives of Life Sciences year 2022 are, starting from third year, Vikram Kamlesh Sehrawat and Manika Chauhan. CR of second year is Ansh Sharma. CR of first year are Rishita Gupta and Nitin Jangra. Your categorization and love for systematizing have really helped us in keeping with organizing and do not let us succumb ourselves with tedious long hour of lab and class period. You have really turned out to be an asset by being a class representative. Now, I would like to call Secretary of our society, Ms. Arushi Dogra, to take the event further.
Following up, I would take a moment to invite our respected, renowned, and generous life science coordinator, Dr. Iranbam Rocky Mangangcha sir, and our assiduous president, Vikram Kamlesh Sehrawat, along with all the office bearers for oath taking ceremony. Over to you, Rocky sir.
Thank you, Arushi. Are you all there? Office
Yes,
bearers?
sir.
Yes,
Okay.
sir. I request all the office bearers and council members to kindly switch on their cameras.
Okay. Shall I start?
Yes, sir.
Okay. Please repeat after me. I,
Vikram Sehrawat,
I, Ansh Sharma,
I, Arushi Dogra,
I, Samaira Bhutani,
I, Malika Chauhan,
I, Ankur Bhardwaj,
I, Mahi Verma,
I, Nyashi Srivastava.
okay. Please unmute, all of you, please unmute, and you can say at the same time also. Okay. Do swear in the name of God.
do swear in the name of God
do swear in the name of God.
do swear
do swear
Hello.
in the name of God
Sir,
Do swear.
I'll be repeating after you.
Okay, then that will be better because like I'm not able to.
Yeah. Yes, sir. I'll be repeating after you. They all will take oath. Yes, sir.
Okay, okay then. So we'll start from the beginning then. I,
Vikram Sehrawat,
do swear in the name of God
do swear in the name of God
that I will faithfully execute the office of President
that I will faithfully execute the office of President
of Synergia
of Synergia
and will with the best of my ability
with best of my abilities
preserve, protect, and defend
will preserve, protect, and defend
the motto of this society,
the motto of this society,
that is to create a vital platform to enable the budding life scientists to have the broader view of applying three disciplines: botany, chemistry, and zoology together in the field of research development, education, industry, and entrepreneurship. And that I will devote myself to the service and well-being of the students of Life Sciences, Deshbandhu College. Okay. All the very best.
Thank you, sir.
Thank you, everyone. It is to inform that feedback form has been posted in the chat box. As rightly said, unity is strength. Where there is teamwork and collaboration, wonderful things can be achieved. With this, we come to the successful ending of the event. I would like to congratulate the team of Synergia for organizing a prosperous event and would like to thank our esteemed guests and wonderful audience to give us their precious time and grace the occasion. I would like to invite our inspiring and untiring convener from Chemistry Department, Dr. Deepti Gupta ma'am, for final vote of thanks. Over to you, ma'am.
Thank you, Arush. Thank you, everyone. Well, I take this opportunity on behalf of whole Life Sciences Society to thank everyone over here for attending the lecture and for attending the whole event, first of all. Then I would also like to obviously extend a hearty vote of thanks to our honorable speaker, Professor Ruplal sir, for giving an excellent coverage of about microbiome in the field of health, environment, and society. The way he has and our Vice Principal Sir, Professor Kamal Gupta Sir, for his encouragement and enormous cooperation in organizing this event. I would also like to thank our coordinator IQAC, Dr. Aditya Saxena Sir, for all the support he has been giving throughout all these events. Then
Ma'am, is there any issue with my network or
No, I think
ma'am's
ma'am
network?
side, yeah.
No, actually, yeah. Even we are not able to listen to her.
I think there's some network glitch.
I'm sorry, my laptop got stuck. Am I still audible?
Yes, you are audible. Yes.
Yeah, I'm so sorry for the technical glitch. It happens when you're about to speak and suddenly your laptop gets stuck somewhere. So should I start again? I don't know where the pause came up. So I'll just start with my vote of thanks again.
You can start again.
Yes. Okay, thank you so much. Thank you so much, Vikram. So I would like to extend a hearty vote of thanks to our honorable speaker, Professor Ruplal sir, for giving an excellent coverage of about microbiome in the field of health, in the sense the way he has explained us the microbes which are there in our gut, in the field of environment, the work he has been doing in this Manikaran and in Khir Ganga, and in the field of society, in which he has enlightened us that how we are neglecting the concept of microbiomes. So it was a brilliant lecture, and I hope everyone over here who have attended the lecture have learned and have gained some knowledge about microbiomes. Now, I would like to extend my vote of thanks to our very dynamic and very encouraging Principal Sir, Professor Rajiv Agarwal Sir, our Vice Principal Sir, who has always been very cooperative in organizing these events. I would also like to extend my thanks to IQAC coordinator, Dr. Aditya Saxena Sir, for his immense support and encouragement. Then I would like to thank coordinator, Life Sciences Society, Dr. Rocky Manjanda Sir, for his guidance in organizing this event. Then, of course, the turn of conveners and co-conveners comes. So I don't want to repeat the names and whole conveners, all the conveners and co-conveners. I would like to extend my hearty thanks to all of you. Last but not the least, the whole team of students, we are very fortunate enough to be backed by the team of dedicated and motivated students who know their job so well and who are always so result oriented. And I especially Vikram and Arushi and Ansh, all these students have been really working hard. Manika, all these students have been really working hard throughout day and night to make these events successful and memorable. And of course, I forgot, I would like to thank our technical support, Dr. Monica Bajaj ma'am, who is from Computer Science Department, and she has always been supporting us in organizing this Zoom platform, virtual conferences and events. And without her support, we would not have been able to do virtual meetings. Thank you so much, ma'am. And thank you everyone for being with us throughout the event. Thank you. Thank you very much.
Thank you, ma'am. Thank you, ma'am. At last, I would like to request everyone present here, please turn on your camera. We want to take some screenshots. All council members, all participants, all teachers, you are kindly requested to turn on your camera. A group picture, Kamal sir's favorite. Everyone, please on your camera. I again request all of you to turn on your camera. And Ansh, please make sure take a good screenshot. Thank you so much, everyone.
Thank you.
We have taken the screenshot. Thank you all of you for being here. And feedback form is there in the chat box. Kindly fill it. And is there any issue with the certificate, please do let us know. Teachers can contact Dr. Manisha Sengar ma'am, and students can contact either me or Arushi.
Thank you, everyone.
Thank you.
Thank you so much.
Congratulations, everyone. Great event.
Thank
Very well
you,
organized.
Thank you.
Thank you so much.
Thank you, sir.
everyone.
Thank you so much, sir. Your inputs are always so valuable.
Yes, sir. Rightly said. Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Many congratulations to everyone.
Thank you.
Same to you. Thank you.
Take care.
Manisha
हाँ बोलो बेटा.
ma'am, with your due permission, can we officially conclude the
Yes
event?
बेटा. Yes, please.
Yeah.
Okay.
Manika, please end the event. Thank you, everyone. Thank you.